Hello, everyone. Thank you very much for joining the presentation, and also thank you for the JCVI Institute for allowing this time to present this information that we published recently this year. Um, we're going to talk about maximizing the biological efficiency, but I like to start by setting the right context. So, understanding the biological efficiency is crucial for exploring life's boundaries, optimizing biotechnological processes, and proving the potential for life beyond Earth. And you may don't know about this already, but your research is already contributing to sustainability and space exploration at the moment. But let me develop and reach this point. So first things first, I'm going to explain some key concepts and walk you through how this project started and how it all developed to its potential applications. So this project started in the astrobiology lab with Dr. Charles Cockell. And for that, we need to understand what habitability is. How is an environment habitable? Well, habitability is the ability of an environment to support activity of at least one known organism. But what do we need for that? We all know this. We need water. We need the right biomolecules. With that, we need the right physical chemical conditions. And we also need, very importantly, energy. But how much energy do we need to have an environment that is habitable? Well, the energy needs to equal or exceed the energetic cost associated with the synthesis and the maintenance of the organism. But what is this minimum energy? What is the threshold energy that allows life? Well, first of all, we need to define the minimum energy necessary, which is pretty much just the sum of all the components, aka building blocks like amino acids, nucleic acids, etc., into their biomolecules, protein, RNA, lipids, etc. And why is this relevant at all? Well, because we can understand the origins of life. We can understand where life is originated here and in other parts of the universe. We can sustain life in different environments, but more importantly, we can understand the minimum requirements, and these allow us to understand the components and the processes that are necessary for the survival and reproduction of an organism, which we already know about this. But more importantly, it allows us to optimize these processes. So again, just to set the right context, I'm going to go super quickly about why we use the Gibbs free energy. So we know there are two types of reactions that require energy or that give energy back. We're interested in the endergonic type of reaction, which requires energy. So how much energy do we need to convert our reactants or building blocks into our products? Well, we all know that the energy will depend basically on two groups of factors, external and internal. External will be temperature, pH, pressure, etc. And internals are going to be product mass. How much proteins does my cell have? How much nucleic acids and amino acids does my cell have to produce proteins? And very importantly, the metabolic pathways. But now, since this difference of energy between reactants and products that gives free energy that we were looking for may vary according to these external factors, we decided to look for the absolute minimum possible energy necessary. And then we decided to ignore some of these factors, particularly metabolic pathways, and just focus on what is the energy taken from point A to point B, reactants to products. And so earlier this year, we published the results, and these are the values we obtained. This is the minimum energy necessary to synthesize a cell of E. coli. All of this at 298 Kelvin. S. cerevisiae, an average mammalian cell, and the JCVI SYN3A cell. As you can see, the lowest value here is the JCVI SYN3A. So Does that give us any insight about whether mass is directly proportional or the complexity of a cell is directly proportional to the energy required and therefore efficiency for an organism? Well, sort of, yes. Um, Let me super quickly tell you how we developed the algorithm. This algorithm, which is publicly available for everyone to try here and calculate the energy any cell you may have. You only are going to require the cell type because every cell type will have a different concentration pool for reactants and products, a genome sequence and a protein sequence because the sequence gives some energetic variability, although uh, small, important, and a temperature, just for the sake to grasp the reality. The Once having the data input, the algorithm will develop a virtual cell, will transcribe the genome sequence, adjust the concentration, and calculate the formation, reaction, and Gibbs free energy of each one of the biomolecules of the virtual cell. And thus we get an average value for the genome, transcriptome, protein, and membrane. Now, the results look something like this. Now, I think it might be a bit hard for you to see the graphs right here. So I went ahead and simplified the information. Here we have three data sets grouped in an average. So I just grabbed the average values we got for E. coli, cerevisiae, mammalian cell, and the SYN3A cell. And just created this charts, pie charts here. So let's go for the first one, average mass fraction. So um, proteins take for most of the cells dry weight, up to 60%. Lipids account for around 15%, glycan 7%, and so on. DNA and RNA are the lowest, DNA being 1% and RNA around 4%. Again, these are just averages. These are not accurate numbers. On the second data set, we have how much energy is required to synthesize 1 gram of each one of those biomolecules. So if we have a cell which has 1, 2, 3, 4, 5 grams of those biomolecules, meaning 1 gram of protein, 1 gram of lipids, and so on, we're going to see what is the energetic fraction that a cell needs to synthesize. We can see protein go down to 21%, and this is probably due because of the concentration pools. Lipids are one of the most expensive biomolecules, probably because of their complexity. Glycans 20%, and this is an important one. DNA is more is more expensive to synthesize than RNA. However, when we go and adjust the biomolecules, the biomolecules to the mass fraction, we're going to see that DNA goes lower than RNA. Proteins is almost a direct correlation, probably again because of the concentration pools, and lipids around 32%. So now, what about comparing the cell types? Well, obviously, mammalian is going to be the most expensive because of its complexity and the amount of biomolecules that it has. Cerevisiae is definitely more expensive than to synthesize than E. coli and JCBI's cell. But here's where it starts getting interesting. So when we compare in our model, the energy necessary to synthesize one E. coli cell and compare it to the SYN3A cell, we can see that the difference is significant. JCBI is significantly more, is cheaper to produce, to synthesize for a cell than E. coli. However, as I mentioned before, the external factors are going to change this considerably. So as I mentioned before, we did not consider the metabolic pathways because we were in search of the minimum, the absolute thermodynamical minimum. So what happens when we consider the metabolic pathways? Well, the story changed a bit. Here is a comparison of 3 studies, ours and one done by McCollum and Ammons on the left. You can see this is the E. coli value, which is significantly higher than the one we obtained, in around 40 ATP millimole of ATP per gram of dry weight, and our value for E. coli is around 10 millimole of ATP. JCVI in our model is slightly less than E. coli in terms of ATP, But comparing to the study to the value reported in Brewer 2019 for the essential metabolism of a minimal cell, we can see that the value reported is slightly higher. Here is a better view of both values. And now, what does it mean? This means that there is a point of reference. Of course, we need to remember that our theoretical value is just that, a theoretical value. And whether it's reachable or not, well, that depends on many factors, as I mentioned, but more importantly, the metabolic pathways. So although it might be very hard to reach this metabolical minimum, at least we have a guidance for us to understand how we can reach to that thermodynamical minimum through modifying metabolic pathways. And so in this case, for example, we will have a biological efficiency of 37% compared to the absolute minimum thermodynamical minimum. And that gives us a pathway on how to proceed in modifying and engineering some metabolic pathways. Now, let's remember there are different thermodynamical minimums. So even the one we obtain thermodynamically can be a different value depending on the condition. So it doesn't mean that we're not close, but it's at least a guidance we can follow for different cell types. Now, what are the future steps? Well, we have now a minimal cell that can serve us as a base to build up upon. So we can start engineering hydrogen metabolic pathways if we already know how far the current energetic cost is to the thermodynamic minimum. And we can engineer all of these pathways to be more efficient with glycolysis, fatty acids, amino acids, you name it. I just put a couple of examples in here. And this, in theory, can give us more efficient biofactories. And more efficient biofactories can give us, well, the tools to be fully sustainable. We can arrange here a cycle of resource to give us energy, food, biofuels, etc. And then the sky is the limit from there. So this is how the minimal cell can contribute to these areas, and that's it. I think I'm on time. So thank you for your time.
Thank you, Edwin. Edwin, I don't understand how you get such a 2.5-fold difference between your calculations and the Luthi-Schulten, the paper I was on, the 2019 eLife paper, Marion Brewer, the first author, how you have such different values for the amount of biological efficiency. There.
Here. Yeah. Well, again, the main take here is that we did not consider the metabolic pathways. Okay, I saw in the paper in 2019, they were doing the calculations following some metabolic pathways, and
but there is no amino acid metabolism, so it's just assimilating. I mean, it's still just putting together proteins from amino acids that are imported, and both of you have to make the same amount of proteins, I would think.
Yeah, that is correct. And that is exactly what I wanted to exemplify here, because again, there are several set of conditions that could be affecting the energetic cost at the end of the day. We decided to go for the absolute minimum. So we did not consider many things that probably were considered in the calculations done in Brewer 2019. So that could be the difference.
So you're just considering the amount of energy needed to do protein translation, not to produce the amino acids?
So instead of the protein translation, it will be protein assembly. So just going from point A, which is amino
translation
acids.
is sort of assembling amino acid subunits.
Yeah, my point with not the translation is I do not consider which enzymes could be involved, for example. I do not
Okay.
consider which pathway is going to follow. I'm just going to consider assembly of amino acids to the final shape of the protein, which is it could be a lot of steps involved in between.
Okay. Other questions for Edwin?
Yeah, perhaps a question that is connected to this one. Do you consider maintenance energy as a factor that contributes to loss of ATP?
All right, that's a good question that I forgot to disclose. We do not consider maintenance energy. We're just considering here synthesis energy, because maintenance energy is a whole different topic that it's going to give different sets of energies based on the conditions as well. But although we could consider synthesis snapshots of the maintenance, there are different processes that are not involved in the synthesis and that are involved in the maintenance. So maintenance energy will be different. Here, we're just considering the synthesis energy, which is what is the minimum threshold of energy required for an environment to provide energy to build the biomolecule necessary to have a living cell.
But wouldn't that then already explain perhaps the 2.5-fold difference with the eLife paper?
Sorry to say that again.
Couldn't that then already explain the large difference between your paper and the 2019 eLife paper? Because I think, especially for minimal cells, the amount of maintenance energy, for instance, futile cycles, leakage across the membrane, is a significant factor.
Yeah, 100%. I agree with you. That's what I meant with the metabolic pathways, because at the end of the day, that's sort of part of the maintenance aspect of a living cell, and that's what I meant. This is just a snapshot of going from point A to point B, so from amino acids to proteins, without considering how to reach there.
Okay, thank you.